The retina is the third and inner coat of the eye, which is a light-sensitive layer of tissue. The optics of the eye create an image of the visual world on the retina, through the cornea and lens, which serves much the same function as the film in a camera. Light striking the retina initiates a cascade of chemical and electrical events that ultimately trigger nerve impulses. These are sent to various visual centers of the brain through the fibers of the optic nerve. Neural retina typically refers to three layers of neural cells: photoreceptor cells, bipolar cells, and ganglion cells, within the retina, while the entire retina refers to these three layers plus a layer of pigmented epithelial cells. In vertebrate embryonic development, the retina and the optic nerve originate as outgrowths of the developing brain, specifically the embryonic diencephalon. Thus, the retina is considered part of the central nervous system, CNS, and is actually brain tissue. It is the only part of the CNS that can be visualized non-invasively. The retina is a layered structure with several layers of neurons interconnected by synapses. The only neurons that are directly sensitive to light are the photoreceptor cells. For vision, these are of two types, the rods and cones. Rods function mainly in dim light and provide black and white vision, while cones support the perception of color. A third type of photoreceptor, the photosensitive ganglion cells, is important for entrainment and reflexive responses to the brightness of light. Neural signals from the rods and cones undergo processing by other neurons of the retina. The output takes the form of action potentials in retinal ganglion cells whose axons form the optic nerve. Several important features of visual perception can be traced to the retinal encoding and processing of light. The term is from Latin rte, meaning net. Structure. The vertebrate retina has 10 distinct layers. From closest to farthest from the vitreous body, that is, from closest to the front exterior of the head towards the interior and back of the head. Inner limiting membrane, basement membrane elaborated by molar cells. Nerve fiber layer, axons of the ganglion cell nuclei. Note that a thin layer of molar cell footplates exists between this layer and the inner limiting membrane. Ganglion cell layer contains nuclei of ganglion cells, the axons of which become the optic nerve fibers for messages and some displaced amacrine cells. Inner plexiform layer contains the synapse between the bipolar cell axons and the dendrites of the ganglion and amacrine cells. Inner nuclear layer contains the nuclei and surrounding cell bodies, perikarya, of the amacrine cells, bipolar cells, and horizontal cells. Outer plexiform layer projections of rods and cones ending in the rod spherule and cone pedicle, respectively. These make synapses with dendrites of bipolar cells. In the macular region, this is known as the fiber layer of Henle. Outer nuclear layer cell bodies of rods and cones. External limiting membrane layer that separates the inner segment portions of the photoreceptors from their cell nucleus. Layer of rods and cones, layer of rod cells and cone cells. Retinal pigment epithelium, single layer of cuboidal cells, with extrusions not shown in diagram. This is closest to the choroid. These can be simplified into four main processing stages: photoreception, transmission to bipolar cells, transmission to ganglion cells which also contain photoreceptors, the photosensitive ganglion cells, and transmission along the optic nerve. At each synaptic stage there are also laterally connecting horizontal and amacrine cells. The optic nerve is a central tract of many axons of ganglion cells connecting primarily to the lateral geniculate body, a visual relay station in the diencephalon, the rear of the forebrain. It also projects to the superior colliculus, the suprachiasmatic nucleus, and the nucleus of the optic tract. It passes through the other layers, creating the optic disc in primates. Additional structures, not directly associated with vision, are found as outgrowths of the retina in some vertebrate groups. In birds, the pecten is a vascular structure of complex shape that projects from the retina into the vitreous humor. It supplies oxygen and nutrients to the eye, and may also aid in vision. Reptiles have a similar, but much simpler, structure. In adult humans, the entire retina is approximately 72% of a sphere, about 22 mm in diameter. The entire retina contains about 7 million cones and 75 to 150 million rods. The optic disc, a part of the retina sometimes called the blind spot because it lacks photoreceptors, is located at the optic papilla, a nasal zone where the optic nerve fibers leave the eye. It appears as an oval white area of 3 mm superscript 2. Temporal, in the direction of the temples, to this disc is the macula. At its center is the fovea, a pit that is responsible for our sharp central vision but is actually less sensitive to light because of its lack of rods. Human and non-human primates possess one fovea as opposed to certain bird species such as hawks who are bifoveate and dogs and cats who possess no fovea but a central band known as the visual streak. Around the fovea extends the central retina for about 6 mm and then the peripheral retina. The edge of the retina is defined by the ora serrata. The length from one ora to the other, or macula, the most sensitive area along the horizontal meridian is about 32 mm. In section the retina is no more than 0.5 mm thick. It has 3 layers of nerve cells and 2 of synapses, including the unique ribbon synapse. The optic nerve carries the ganglion cell axons to the brain and the blood vessels that open into the retina. The ganglion cells lie outermost in the retina while the photoreceptive cells lie innermost. Because of this counterintuitive arrangement, light must first pass through and around the ganglion cells and through the thickness of the retina, including its capillary vessels, not shown, before reaching the rods and cones. However, it does not pass through the retinal pigment epithelium or the choroid, both of which are opaque. The white blood cells in the capillaries in front of the photoreceptors can be perceived as tiny bright moving dots when looking into blue light. This is known as the blue field entoptic phenomenon, or Shearer's phenomenon. Between the ganglion cell layer and the rods and cones there are 2 layers of neuropils where synaptic contacts are made. The neuropil layers are the outer plexiform layer and the inner plexiform layer. In the outer the rods and cones connect to the vertically running bipolar cells, and the horizontally oriented horizontal cells connect to ganglion cells. The central retina is cone-dominated and the peripheral retina is rod-dominated. In total there are about 7 million cones and 100 million rods. At the center of the macula is the foveal pit where the cones are smallest and in a hexagonal mosaic, the most efficient and highest density. Below the pit the other retina layers are displaced, before building up along the foveal slope until the rim of the fovea or parafovea, which is the thickest portion of the retina. The macula has a yellow pigmentation from screening pigments and is known as the Macula Lutea. The area directly surrounding the fovea has the highest density of rods converging on single bipolars. Since the cones have a much lesser power of merging signals, the fovea allows for the sharpest vision the eye can attain. Though the rod and cones are a mosaic of sorts, transmission from receptors to bipolars to ganglion cells is not direct. Since there are about 150 million receptors and only 1 million optic nerve fibers, there must be convergence and thus mixing of signals. Moreover, the horizontal action of the horizontal and amacrine cells can allow one area of the retina to control another, e.g. one stimulus inhibiting another. This inhibition is key to the sum of messages sent to the higher regions of the brain. In some lower vertebrates, e.g. the pigeon, there is a centrifugal control of messages, that is, one layer can control another, or higher regions of the brain can drive the retinal nerve cells, but in primates this does not occur. Development. Retinal development begins with the establishment of the eye fields mediated by SHH and SIX3 with subsequent development of the optic vesicles via PAX6 and LHX2. The role of PAX6 in eye development was elegantly demonstrated by Walter Gehring and colleagues, who showed that ectopic expression of PAX6 can lead to eye formation on Drosophila antennae, wings, and legs. The optic vesicle gives rise to 3 structures: the neural retina, the retinal pigmented epithelium, and the optic stalk. The neural retina contains the retinal progenitor cells, RPCs, that give rise to the 7 cell types of the retina. Differentiation begins with the retinal ganglion cells and concludes with production of the molar glia. Although each cell type differentiates from the RPCs in a sequential order, there is considerable overlap in the timing of when individual cell types differentiate. The cues that determine a RPC daughter cell fate are coded by multiple transcription factor families including the BHLH and homeodomain factors. In addition to guiding cell fate determination, cues exist in the retina to determine the dorsal/ventral and nasal/temporal axes. The DV axis is established by a ventral to dorsal gradient of VAX2, whereas the NT axis is coordinated by expression of the forkhead transcription factors FOXD1 and FOXG1. Additional gradients are formed within the retina that aid in proper targeting of RGC axons that function to establish the retinotopic map. Blood supply. The retina is stratified into distinct layers, each containing specific cell types and/or cellular compartments. Moreover, each layer has unique metabolic requirements. To accommodate this differential metabolic demand, the ophthalmic artery bifurcates and supplies the retina via two distinct vascular networks. The choroidal network, which supplies the choroid and the outer retina, and the retinal network, which supplies the retina's inner layer. The topology of the inner retinal vasculature is known to conform to network principles that are related to certain physical properties. The unique structure of the blood vessels in the retina has been used for biometric identification. Changes in the retinal microcirculation are seen with aging, exposure to air pollution, and may indicate cardiovascular diseases such as hypertension and atherosclerosis. The identification of vascular bifurcations is one of the basic steps in this analysis. Results of such analyses of the retinal microcirculation can be evaluated against the ground truth data of vascular bifurcations of retinal fundus images that are obtained from the DRIVE data set. In addition, the classes of vessels of DRIVE dataset has also been identified in. An automated method for accurate extraction of these bifurcations is also available in. Determining the equivalent width of arterioles and venules near the optic disc is also a widely used technique to identify cardiovascular risks. The bird retina is devoid of blood vessels, perhaps to give unobliterated light for forming images, thus giving better resolution. It is, therefore, a considered view that the bird retina depends for nutrition and oxygen supply on a specialized organ, called the pectin or pectin oculi, located on the blind spot or optic disc. This organ is extremely rich in blood vessels and is thought to supply nutrition and oxygen to the bird retina by diffusion through the vitreous body. The pectin is highly rich in alkaline phosphatase activity and polarized cells in its bridge portion, both befitting its secretory role. Pectin cells are packed with dark melanin granules, which have been theorized to keep this organ warm with the absorption of stray light falling on the pectin. This is considered to enhance metabolic rate of the pectin, thereby exporting more nutritive molecules to meet the stringent energy requirements of the retina during long periods of exposure to light. Function. An image is produced by the patterned excitation of the cones and rods in the retina. The excitation is processed by the neuronal system and various parts of the brain working in parallel to form a representation of the external environment in the brain. The cones respond to bright light and mediate high-resolution color vision during daylight illumination, also called photopic vision. The rods are saturated at daylight levels and don't contribute to pattern vision. However, rods do respond to dim light and mediate lower resolution, monochromatic vision under very low levels of illumination, called scotopic vision. The illumination in most office settings falls between these two levels and is called mesopic vision. At these light levels, both the rods and cones are actively contributing pattern information to that exiting the eye. What contribution the rod information makes to pattern vision under these circumstances is unclear. The response of cones to various wavelengths of light is called their spectral sensitivity. In normal human vision, the spectral sensitivity of a cone falls into one of three subgroups. These are often called blue, green, and red cones, but more accurately are short, medium, and long wavelength sensitive cone subgroups. It is a lack of one or more of the cone subtypes that causes individuals to have deficiencies in color vision or various kinds of color blindness. These individuals are not blind to objects of a particular color but experience the inability to distinguish between two groups of colors that can be distinguished by people with normal vision. Humans have three different types of cones: trichromatic vision, while most other mammals lack cones with red sensitive pigment and therefore have poorer, dichromatic, color vision. However, some animals have 4 spectral subgroups, e.g. the trout adds an ultraviolet subgroup to short, medium, and long subgroups that are similar to humans. Some fish are sensitive to the polarization of light as well. When light falls on a receptor, it sends a proportional response synaptically to bipolar cells, which in turn signal the retinal ganglion cells. The receptors are also cross-linked by horizontal cells and amacrine cells, which modify the synaptic signal before the ganglion cells. Rod and cone signals are intermixed and combine, although rods are mostly active in very poorly lit conditions and saturate in broad daylight, while cones function in brighter lighting because they are not sensitive enough to work at very low light levels. Although all are nerve cells, only the retinal ganglion cells and few amacrine cells create action potentials. In the photoreceptors, exposure to light hyperpolarizes the membrane in a series of graded shifts. The outer cell segment contains a photopigment. Inside the cell, the normal levels of cyclic guanosine monophosphate, cGMP, keep the NAND channel open, and thus in the resting state the cell is depolarized. The photon causes the retinal bound to the receptor protein to isomerase to transretinal. This causes receptor to activate multiple G proteins. This in turn causes the GA subunit of the protein to activate a phosphodiesterase, PDE6, which degrades cGMP, resulting in the closing of NAN cyclic nucleotide-gated ion channels, CNGs. Thus the cell is hyperpolarized. The amount of neurotransmitter released is reduced in bright light and increases as light levels fall. The actual photopigment is bleached away in bright light and only replaced as a chemical process, so in a transition from bright light to darkness the eye can take up to 30 minutes to reach full sensitivity, see adaptation, I. In the retinal ganglion cells there are 2 types of response, depending on the receptive field of the cell. The receptive fields of retinal ganglion cells comprise a central approximately circular area, where light has one effect on the firing of the cell, and an annular surround, where light has the opposite effect on the firing of the cell. In on cells, an increment in light intensity in the center of the receptive field causes the firing rate to increase. In off cells, it makes it decrease. In a linear model, this response profile is well described by a difference of Gaussians and is the basis for edge detection algorithms. Beyond this simple difference, ganglion cells are also differentiated by chromatic sensitivity and the type of spatial summation. Cells showing linear spatial summation are termed X cells, also called parvocellular, P, or midget ganglion cells, and those showing nonlinear summation are Y cells, also called magnocellular, M, or parasol retinal ganglion cells, although the correspondence between X and Y cells, in the cat retina, and P and M cells, in the primate retina, is not as simple as it once seemed. In the transfer of visual signals to the brain, the visual pathway, the retina is vertically divided in 2, a temporal, nearer to the temple, half, and a nasal, nearer to the nose, half. The axons from the nasal half cross the brain at the optic chiasma to join with axons from the temporal half of the other eye before passing into the lateral geniculate body. Although there are more than 130 million retinal receptors, there are only approximately 1.2 million fibers, axons, in the optic nerve. A large amount of pre-processing is performed within the retina. The fovea produces the most accurate information. Despite occupying about 0.01% of the visual field, less than 2 degrees of visual angle, about 10% of axons in the optic nerve are devoted to the fovea. The resolution limit of the fovea has been determined at around 10,000 points. See visual acuity. The information capacity is estimated at 500,000 bits per second. For more information on bits, see information theory, without color, or around 600,000 bits per second including color. Spatial encoding. The retina does not simply send a picture to the brain. The retina spatially encodes, compresses, the image to fit the limited capacity of the optic nerve. Compression is necessary because there are 100 times more photoreceptor cells than ganglion cells as mentioned above. The retina does so by decorrelating the incoming images in a manner to be described below. These operations are carried out by the center surround structures as implemented by the bipolar and ganglion cells. There are two types of center surround structures in the retina: on centers and off centers. On centers have a positively weighted center and a negatively weighted surround. Off centers are just the opposite. Positive weighting is more commonly known as excitatory and negative weighting is more commonly known as inhibitory. These center surround structures are not physical in the sense that one cannot see them by staining samples of tissue and examining the retina's anatomy. The center surround structures are logical, i.e., mathematically abstract, in the sense that they depend on the connection strengths between ganglion and bipolar cells. It is believed that the connection strengths between cells is caused by the number and types of ion channels embedded in the synapses between the ganglion and bipolar cells. See receptive field for figures and more information on center surround structures. The center surround structures are mathematically equivalent to the edge detection algorithms used by computer programmers to extract or enhance the edges in a digital photograph. Thus the retina performs operations on the image to enhance the edges of objects within its visual field. For example, in a picture of a dog, a cat, and a car, it is the edges of these objects that contain the most information. In order for higher functions in the brain, or in a computer for that matter, to extract and classify objects such as a dog and a cat, the retina is the first step to separating out the various objects within the scene. As an example, the following matrix is at the heart of the computer algorithm that implements edge detection. This matrix is the computer equivalent to the center surround structure. In this example, each box, element, within this matrix would be connected to one photoreceptor. The photoreceptor in the center is the current receptor being processed. The center photoreceptor is multiplied by the +1 weight factor. The surrounding photoreceptors are the nearest neighbors to the center and are multiplied by the -1/8 value. The sum of all 9 of these elements is finally calculated. This summation is repeated for every photoreceptor in the image by shifting left to the end of a row and then down to the next line. The total sum of this matrix is 0 if all the inputs from the 9 photoreceptors are the same value. The 0 result indicates the image was uniform, non-changing, within this small patch. Negative or positive sums mean something was varying, changing, within this small patch of 9 photoreceptors. The above matrix is only an approximation to what really happens inside the retina. The differences are: the above example is called balanced. The term balanced means that the sum of the negative weights is equal to the sum of the positive weights so that they cancel out perfectly. Retinal ganglion cells are almost never perfectly balanced. The table is square while the center surround structures in the retina are circular. Neurons operate on spike trains traveling down nerve cell axons. Computers operate on a single floating point number that is essentially constant from each input pixel. The computer pixel is basically the equivalent of a biological photoreceptor. The retina performs all these calculations in parallel while the computer operates on each pixel one at a time. There are no repeated summations and shifting as there would be in a computer. Finally, the horizontal and amacron cells play a significant role in this process, but that is not represented here. Here is an example of an input image and how edge detection would modify it. Once the image is spatially encoded by the center surround structures, the signal is sent out the optical nerve, via the axons of the ganglion cells, through the optic chiasm to the LGN, lateral geniculate nucleus. The exact function of the LGN is unknown at this time. The output of the LGN is then sent to the back of the brain. Specifically, the output of the LGN radiates out to the V1 primary visual cortex. Simplified signal flow, photoreceptors bipolar ganglion chiasm LGN V1 cortex. Clinical significance. There are many inherited and acquired diseases or disorders that may affect the retina. Some of them include: retinitis pigmentosa is a group of genetic diseases that affect the retina and cause the loss of night vision and peripheral vision. Macular degeneration describes a group of diseases characterized by loss of central vision because of death or impairment of the cells in the macula. Rod Dystrophy, CORD, describes a number of diseases where vision loss is caused by deterioration of the cones and/or rods in the retina. In retinal separation, the retina detaches from the back of the eyeball. Ignopuncture is an outdated treatment method. The term retinal detachment is used to describe a separation of the neurosensory retina from the retinal pigment epithelium. There are several modern treatment methods for fixing a retinal detachment: pneumatic retinopexy, scleral buccal, cryotherapy, laser photocoagulation, and pars plana vitrectomy. Both hypertension and diabetes mellitus can cause damage to the tiny blood vessels that supply the retina, leading to hypertensive retinopathy and diabetic retinopathy. Retinoblastoma is a cancer of the retina. Retinal diseases in dogs include retinal dysplasia, progressive retinal atrophy, and sudden acquired retinal degeneration. Lipemia retinalis is a white appearance of the retina, and can occur by lipid deposition in lipoprotein lipase deficiency. Diagnosis and treatment. A number of different instruments are available for the diagnosis of diseases and disorders affecting the retina. Ophthalmoscopy and fundus photography are used to examine the retina. Recently, adaptive optics has been used to image individual rods and cones in the living human retina, and a company based in Scotland have engineered technology that allows physicians to observe the complete retina without any discomfort to patients. The electroretinogram is used to measure non-invasively the retina's electrical activity, which is affected by certain diseases. A relatively new technology, now becoming widely available, is optical coherence tomography, OCT. This non-invasive technique allows one to obtain a 3D volumetric or high-resolution cross-sectional tomogram of the retinal fine structure with histologic quality. Treatment depends upon the nature of the disease or disorder. Transplantation of retinas has been attempted, but without much success. At MIT, the University of Southern California, RWTH Auckland University, and the University of New South Wales, an artificial retina is under development, an implant which will bypass the photoreceptors of the retina and stimulate the attached nerve cells directly, with signals from a digital camera. Retinal gene therapy. Gene therapy holds promise as a potential avenue to cure a wide range of retinal diseases. This involves using a non-infectious virus to shuttle a gene into a part of the retina. Recombinant adeno-associated virus, RAAV, vectors possess a number of features that render them ideally suited for retinal gene therapy, including a lack of pathogenicity, minimal immunogenicity, and the ability to transduce post-mitotic cells in a stable and efficient manner. RAAV vectors are increasingly utilized for their ability to mediate efficient transduction of retinal pigment epithelium, RPE, photoreceptor cells, and retinal ganglion cells. Each cell type can be specifically targeted by choosing the appropriate combination of AAV serotype, promoter, and intraocular injection site. Several clinical trials have already reported positive results using RAAV to treat Leber's congenital amaurosis, showing that the therapy was both safe and effective. There were no serious adverse events, and patients in all three studies showed improvement in their visual function as measured by a number of methods. The methods used varied among the three trials, but included both functional methods such as visual acuity and functional mobility, as well as objective measures that are less susceptible to bias, such as the pupil's ability to respond to light, and improvements on functional MRI. Improvements were sustained over the long term, with patients continuing to do well after more than 1.5 years. The unique architecture of the retina and its relatively immune-privileged environment help this process. Tight junctions that form the blood-retinal barrier separate the subretinal space from the blood supply, thus protecting it from microbes and most immune-mediated damage, and enhancing its potential to respond to vector-mediated therapies. The highly compartmentalized anatomy of the eye facilitates accurate delivery of therapeutic vector suspensions to specific tissues under direct visualization using microsurgical techniques. In the sheltered environment of the retina, AAV vectors are able to maintain high levels of transgene expression in the retinal pigmented epithelium, RPE, photoreceptors, or ganglion cells for long periods of time after a single treatment. In addition, the eye and the visual system can be routinely and easily monitored for visual function and retinal structural changes after injections with non-invasive advanced technology, such as visual acuities, contrast sensitivity, fundus autofluorescence, FAF, dark adapted visual thresholds, vascular diameters, pupillometry, electroretinography, ERG, multifocal ERG, and optical coherence tomography, OCT. This strategy is effective against a number of retinal diseases that have been studied, including neovascular diseases that are features of age-related macular degeneration, diabetic retinopathy, and retinopathy of prematurity. Since the regulation of vascularization in the mature retina involves a balance between endogenous positive growth factors, such as vascular endothelial growth factor, VEGF, and inhibitors of angiogenesis, such as pigment epithelium-derived factor, PEDF, RAV-mediated expression of PEDF, angiostatin, and the soluble VEGF receptor SFLT1, which are all anti-angiogenic proteins, have been shown to reduce aberrant vessel formation in animal models. Since specific gene therapies cannot readily be used to treat a significant fraction of patients with retinal dystrophy, there is a major interest in developing a more generally applicable survival factor therapy. Neurotrophic factors have the ability to modulate neuronal growth during development to maintain existing cells and to allow recovery of injured neuronal populations in the eye. AAV encoding neurotrophic factors such as fibroblast growth factor, FGF, family members and GDNF either protected photoreceptors from apoptosis or slowed down cell death. Society and culture. Retinal scan. A retinal scan can be used as a method of biometric identification as the pattern of blood vessels in the retina is unique to an individual. History. In 1894, Santiago Ramón y Cajal published the first major characterization of retinal neurons in Retina der Wirbeltiere, the retina of vertebrates.
Please subscribe and thanks for watching.